This everyone. This is the March 2024 Statistics Former Student, Statistics Former Student Network webinar. We're very fortunate to have with us today Dr. Mandy Herring. Mandy is a professor in the Department of Statistical Science at Baylor University. She obtained her PhD in 2009 in statistics from Texas A&M University under the advisement of Mark Ginton. She spent the next 7 years as an assistant and then associate professor at Colorado School of Mines before joining Baylor. She has over 60 peer-reviewed papers in both statistics and subject matter journals. Her current research focuses on developing methods to solve problems in natural and engineered water environments using data. She uses statistical methods such as time series, functional data analysis, multivariate analysis, and modeling. She's an associate editor for the journals Environmetrics, Technometrics, and Data Science and Science. Sorry, I'm having trouble talking today. She has been selected for early investigator awards from both the International Environmetrics Society and the American Statistical Association section on statistics in the environment. She has also received over $3 million to support her work through organizations such as the National Science Foundation and Department of Energy. Mandy will talk to us today on multivariate statistical process monitoring and complex water and wastewater treatment systems. Take it away, Mandy. Thank you.
Thank you, Marcy. I want to thank the Statistics Former Student Network for inviting me to give this talk. It's fun to be back virtually at A&M and kind of reflect a little bit on some of the work that I've done since graduating and how being at Texas A&M has sort of shaped my career. And so I want to thank Elaine and Anna and Marcy for organizing and putting this together. And just to start out, I always like to start with acknowledgement, so I don't forget people at the end, or I don't run out of time at the end. But over the last few years, I've worked with a lot of amazing faculty and students who have made a lot of this work come alive. And my primary collaborator is with Zahi Kath. Zahi is a full professor at Colorado School of Mines in the Civil and Environmental Engineering department there. And every applied or methodological statistician, I feel like, should have a Zahi, because Zahi is truly amazing. He has he builds lots and lots of water and wastewater treatment systems and outfits them with SCADA systems that collect data, and then brings the data to me and says, what can we do with this? And how can we make our operation of these systems better? So with Zahi, he and I have been funded by the National Science Foundation, the U.S. Department of Energy, the U.S. Bureau of Reclamation, and some other sources. And this is a smattering of former students, current students, postdocs, and other faculty who have helped us with a lot of these different projects, who've contributed to many of these projects. And I know I've left someone out, so if I have, I'm sorry. This is a little bit about me, just beyond what Marcy gave in terms of introduction. I was born in Louisiana and was there for 18 years. And then I moved to Waco here at Baylor and did my undergraduate degree here at Baylor. After Baylor, I moved to Montana, and I spent a couple of years there getting a master's degree in statistics at Montana State. Then I moved back to College Station and that's when I did my PhD at A&M under the advisement of Mark Ginton, who's now at KAUST. And then I took my first faculty job at Colorado School of Mines in Golden and went from assistant to associate professor there, and then moved back to Waco to take an associate professor position in the Department of Statistics at Baylor, and now I'm a full professor here. And so I think with this sort of diagram, as we're always looking for patterns, I was telling Elaine, I think this is exactly where I'll probably retire, right up here in North Texas, if I can judge based on the pattern. Some of the work that I've done, that I started out doing when I was at A&M was on wind forecasting and characterization of wind patterns in both space and time. But I've also been interested in lots of different types of applied work, and any kind of interesting application I'm on board with. And so right now, my current work is a lot to do with spatial clustering, monitoring isolation and control, and water related problems, and I have some functional data analysis projects as well. But today I'm going to really focus on these water related problems and monitoring in those areas. Um, just as a why are water and wastewater treatment so important slide, that's what this one is about. We have a National Science Foundation grant, a data science core, which is about workforce education, both among undergraduates, graduates, and also people who are already professionals, to train them in data science. And I didn't realize before I got into this field, even as I was just beginning to get into water and wastewater treatment applications, that annually almost $150 billion is spent on improvement, operating, and maintaining water and wastewater treatment facilities. That's an enormous dollar amount, and yet there's not a big connection between data science and water and wastewater treatment. And that's typically because the industry is really slow to develop. Of course, it's very conservative because water is so important, and any sorts of failures can have huge public and environmental consequences. So, but the problem is that there are lots more demands being placed on water. And so operators are under pressure to reduce their cost while also increasing and improving water quality. So through this big program, we developed an introduction to data science course where we used a lot of water and wastewater treatment applications. And then we took students from that course and solicited them to do a 5-week undergraduate research program that transitioned into a longer 8-week summer internship. And these were all targeted towards students in data science, statistics, and computer science. But then we also targeted graduate students in other disciplines, because especially those who are in environmental engineering or those professionals who are already working in the water and wastewater treatment industry, we wanted to provide them with some tools as well, to incorporate data science into their work. So um, I'll kind of wrap back around to this program at the end, where I'll show where we have a lot of these data sets that we have worked on and collected over the years, that we've now posted in a public repository. So just to re-emphasize, oftentimes it's not that we don't have enough water on the planet, but having it where it's needed and when it's needed is what's challenging. And so this is just a projection of water risks in the year 2050. And you can see that the highest risks tend to be in areas where there's not a lot of water. It tends to be a dry climate, and that coincides with a large population. But it's not always places where it's dry. You'll see here in Florida, there are some very high risk areas, and here in Arkansas as well. So just because a place has a lot of water, or we perceive it to have a lot of water, doesn't mean that there aren't some really big challenges associated with providing the amount of water where it's needed. So there are lots of solutions being developed for improvements in water treatment, and unique treatments, new types of treatment that will be more efficient, that will produce more water. And one of those solutions is on-site water treatment and reuse. So the idea is that you would take water that has been treated by a wastewater treatment facility, treat it further, and then potentially be able to reuse it as opposed to releasing it back into the environment. But a lot of these on-site water treatment facilities are much smaller, and they don't necessarily have an operator who is able to be there on site 24/7 like a traditional water or wastewater treatment facility would have. So they have to be monitored very carefully in order to, as I mentioned before, reduce human or public and environmental health risks. So I'm going to talk about briefly two projects today, and then just show us a smattering of other projects that we've worked on. I know this is a statistics audience, but it's also an online talk, so I'll try to keep lots of pictures in here and give lots of references at the end if you're interested in following up with the specifics and the details. So the two projects that I'll talk about today in more detail are to do with this sequencing batch membrane bioreactor. And so you can see it here in this picture. This is when it was first installed, but today it's housed in this building. So it's a small decentralized facility. And the idea is that we want to be able to take all the data that is being generated by this decentralized facility and be able to detect any sorts of upsets either in the water that's coming into the facility that it's being required to treat, or the system itself. So the operation of the system has a blower failed, has a pipe clogged, those types of things. Those are the kinds of faults that we want to detect. And then once we've detected, just using the online streaming data, that there's a problem, the next step would be to isolate that fault and be able to figure out, given all this data, which variables were the ones that highly contributed to that fault. Because once we can do that, then the operators can diagnose the fault and then go in and potentially replace the pump or unclog the pipe, or add some additional chemical that's needed. But we want to be able to do this remotely so that you don't necessarily have to have an operator on site 24/7. So with every system, and I've worked with Zahi on many different systems that he's built, it takes a long time for me as a statistician to understand all of the ins and outs of the system. And so in this particular one, we have water that comes in and then it spends some time in these sequencing batch reactors. Then from there, for about 2 hours in the sequencing batch reactors, then they move on to these membrane bioreactors, and then cleaner, I won't say completely clean, but much cleaner water will exit the system. And there's a series of pumps and valves and return. Sometimes there's sludge that comes from the membrane bioreactor that's returned back into the sequencing bioreactor. And throughout this entire system, data is being collected. So things like total suspended solids, conductivity of the water, pH. And you can imagine these sensors are in an environment that are not very hospitable. It's really dirty water. And so the sensors can potentially get fouled or they can fail. And so that's another kind of fault that we might want to be able to detect. And so just sort of breaking it down, this is a picture, a top-down picture with one of my former graduate students looking down into the sequencing batch reactor. It's basically a large vat of sludge where water is pushed in, and there's a microbial community there that's responsible for the nitrification of ammonia. And then once the water has spent some time here and nitrification has occurred, then it passes through an MBR, membrane bioreactor, which is a big membrane that's composed of a lot of these fibers that look like really long Q-tips. And this is where the liquids are separated from the water. And under the hood is a SCADA system that you can see here that's collecting and logging all of this data at a 1-minute frequency. It can log faster than that, but that's the frequency at which it's currently being recorded. And so our goal is to develop a method to detect and isolate abnormal system behavior very quickly and accurately so that we can reduce cost with undue stress to the system or minimize operational downtime. There was, I will show a case study in which there was a process upset, and when the biological organisms in the SBR are impacted or killed off, which can happen, then it can take the system months to recover. And so that's certainly a situation you would not want to have repeated. But doing this fault fault detection and process monitoring is particularly challenging from a statistical point of view for many reasons. There's a lot of data being recorded, a lot of variables being recorded. There are often nonlinear relationships among the variables. They're temporally dependent, especially if we're recording every 1 to 10 minutes, logging observations. There's also pretty strong non-stationarity, so there are diurnal changes and seasonal changes that are expected, or they change how the system is operated, and that has some impacts on the data as well. We are not in a world where anything is normally distributed. We can have lots of different kinds of faults, and different methods can be tuned to identify specific types of faults. And there are physical models, so systems of differential equations that might be able to describe these systems, but they're often too complex to analyze in real time, and they tend to be very specific for the kind of system. So you'd have to build a whole new one for every different system that's built. And we have lots of these variables that they don't all need to be monitored. And so we'll talk more about that later. So what is traditional statistical process monitoring? And sometimes I find statisticians call this statistical process control, but having worked with the engineers now for several years, that word control is very, very different for them than what it means for us as statisticians. So process monitoring is just observing the data and identifying when it becomes unusual relative to some reference set. But control means actually manipulating variables in the system. And that's not what we're doing. So I'm very careful to call this statistical process monitoring. And we can think about this process monitoring in terms of univariate versus multivariate monitoring. So in univariate statistical process monitoring, we're just taking one time series of observations. We calculate some mean and then identify, say, 3 standard deviations above and below that mean. And then when the observations exceed that 3 standard deviations above or below, then some alarm is triggered. That's pretty simplistic. It doesn't necessarily work when you've got 50 variables or 30 variables to monitor. It becomes much more difficult. And so we move to multivariate statistical process monitoring, which allows you to monitor a lot of variables at the same time. We can look for changes in the mean of the process and or covariances now. So both the variability of the variables and how they're related in a pairwise fashion to each other. And we can condense information from lots of variables into a couple of monitoring statistics so that instead of having to monitor each variable individually, we can just grab a couple of monitoring statistics and track those. So common methods are things like multivariate exponentially weighted moving average, multivariate Q-sum. These are some common ones that you'll see in the process monitoring literature. And it's also very common to see PCA applied to the data. And that's one of the methods that I'll share with you all today. Right. So this is a little bit of an overview of the process that we use for this particular problem. We're looking to do this in real time, so not offline and not retrospectively. And what we do is we look through all the variables and we sit down with our collaborators and we say, okay, which variables do we want to monitor? These are the ones that we're interested in detecting any kind of changes. But there's another set of variables, which I'll call explanatory variables. These are variables that can change and, in fact, impact the monitoring variables, but they are impacting the monitoring variables in expected ways. So if I increase the set point of the dissolved oxygen, then I expect the dissolved oxygen in the tank to increase. And that would not be a fault that I would want to flag because that's an expected change because the set point has been changed by the operator. So those are the sorts of things that we would classify as explanatory variables, set points of variables and or things that we can't control, such as ambient temperature. So we want to take these explanatory variables and remove their effects from the monitoring variables and then monitor the residuals. And so we do this with a training set, and we've tested several different types of detrending models, machine learning models, time series models, simple regression models. And then we get residuals and we estimate the principal component analysis model from that. And then we can compute monitoring statistics such as t-squared and SPE, which I'll explain. We estimate some thresholds, which we say, okay, if t-squared or SPE, these monitoring statistics are above this value, then issue an alarm. And then once we've done this all in the training set, we come down and we start testing. Right, so we take a new observation, apply this detrending model, apply the PCA model, both of these from the training set, calculate t-squared and SPE, check if it exceeds the threshold from the training set, and then declare that observation as being either in control, or it's normal, it's typical, it's usual, or it's out of control, it's unusual. And so in terms of PCA, that's one way to reduce the dimension of the data. And we, for some reason, this figure always doesn't show up when I put it in full screen mode. But we see here, this is what is called the Swiss roll data set. And when you apply PCA to this three-dimensional data set, it kind of squashes it. So if there are nonlinear relationships in the data, then PCA applied to it just doesn't quite capture it the way you'd want. So we also tested some other approaches to the dimension reduction, thinking that maybe this will help us with any potential nonlinearities that are in the data. So we tried kernel principal component analysis and local linear embedding. There are other options that you can try as well, but you can see that these two approaches capture more of the swirl here, either by squashing the swirl on the side, or unrolling the swirl. I keep saying squirrel. Unrolling the swirl with local linear embedding. Okay. And I'm going to try to reshare my screen. Let me make this full screen. Elaine, when I change slides here, can you still see that? Or Marcy?
Yeah, I saw you make a change. So I think I see that I can see something different now.
Okay, all right,
You're-
great. I just want to make sure that was still working. So we've looked at different approaches to reducing the dimension of the data. We've looked at some various adaptations for addressing temporal dependence and non-stationarity as well. And then once we've done that, we actually calculate these two statistics, SPE and t-squared. So I'm gonna sit here for a minute so I can explain this figure. We can think of A and B as being the original point. So imagine that we have a three-dimensional space, or three monitoring variables, for example. So we have three monitoring variables and we've plotted them in 3D space. And then we've projected them down into 2D space, into this plane with PCA. And so we can see that A has to travel a long way to get down to this two-dimensional space. And this distance squared is SPE. Sometimes it's called Q, but it's the squared prediction error, and it measures the goodness of fit of this lower dimensional model. And then point B doesn't have to travel very far to get to the two-dimensional space, but once it's there, it's really far from the main cloud of points. So it has a large, what we call t-squared here. It's measuring deviations from the center of this point cloud in the lower dimensional space. And so SPE is calculated, again, based on a eigenvalue, eigenvector decomposition of the correlation matrix of the residuals of all the variables that you're monitoring. P sub k is a matrix of the eigenvectors. And usually when you do PCA, you are only capturing or retaining a subset of those eigenvalues or eigenvectors. And so this sub k represents the k eigenvectors that are retained. And this R represents the residuals from the data, from the monitoring variables. And so this is how SPE is calculated. And then T squared, again, here are the residuals of the monitoring variables that we're interested in, the eigenvectors, and this is a matrix with the K eigenvalues on the diagonal. And so both of these have parametric distributions. SPE follows a chi-square distribution. T squared follows an F distribution. These are scaled chi-squared and F distributions, but they're assuming that the data that you have collected is normally distributed for these distributions to hold. And so what we have found is that, for example, here are all of this histogram is a histogram of all of the T squared values calculated on some training set. And what we find is when we overlay the scaled F distribution here, it doesn't quite fit with the observed T squared values. And this is the threshold. So we see that we would actually classify all of these observations as being unusual or out of control. But when we do a non-parametric fit, which is basically a non-parametric density estimator here, it actually pushes that threshold much farther into the upper tail. And so we identify basically fewer false alarms when we do a non-parametric estimation of this threshold. And we've looked also at lots of different ways to estimate these thresholds when you're in situations with strong temporal dependence and different types of sample sizes. So we've actually evaluated not just this particular non-parametric threshold estimator, but several others as well. So we did a simulation study, and in this simulation study, we simulated 3 variables, x1, 2, and 3, and we introduced here in this guy a shift, a downward shift. Here in x1, we introduced a drift. And this is a latent fault that actually affected all 3 variables simultaneously. So we wanted to check and see if we were comparing PCA with kernel principal components, with local linear embedding, with different variations. This one is static, a dynamic, an adaptive, and an adaptive dynamic variation on PCA with those non-parametric thresholds. How quickly would each of these methods detect the fault? And so these numbers over here are the run lengths, the average run length. So on average, it takes about 20 observations for this particular variation of PCA to identify the shift fault. And that's the best one across all of these methods. The drift fault is much more difficult to detect, but again, the PCA with the adaptive dynamic variations detects it after about 400 observations. And then finally, this latent fault gets detected fastest by KPCA with the adaptive dynamic. The adaptive dynamic variation seems to improve all of the methods. It's not too terribly different from using just PCA. So that's why we have sort of landed on using PCA because it's simple to use. The nonlinearity in the system is not so strong that KPCA and LLE really improve results. So we had a fault in the SBMBR, the system that I described at the beginning, that prompted all of this work. And what happened, you'll see here, this is pH, is that pH really had a very strong drop. And at the same time that pH was coming down, conductivity was coming up. And what happened was this, as pH decreased, this is what killed off the microbial community in the SBR. And you can see there's a big jump up here. This is when Zahi added base to try to stabilize the pH. It continued to come down. He added base again, and then he added base a 3rd time. But the key thing here is that they were not aware that there was a problem in the system until right here at this red line. This is when they started to get alarms on the system. Oh my gosh, pH is going down, down, down. Conductivity is coming up. And when we run our adaptive dynamic PCA monitoring approach, we identified a problem in the system almost 3 days before the operators could see it visually. And so that was sort of our first indicator that, hey, we could actually probably do some things here that would really have a benefit for this particular application area. Our next step was to look at fault isolation. So just because there's been a fault identified isn't really helpful. We need to know which variables are contributing to that fault. And so there are a lot of methods out there for fault detection, for process monitoring, but a lot fewer that have been proposed for fault isolation. And when you're thinking about fault isolation, there are even fewer that can handle non-stationarity and autocorrelation in the data. So in this particular second phase of this project, we're going to do some retrospective monitoring. I'll present the retrospective monitoring, and we've also done prospective monitoring. So the retrospective is we've already got a big chunk of data. Let's look back at it and see where we can identify faults and identify their causes. And then the prospective monitoring would be, let's do this in real time. So again, our method does a step initially where we detrend the data, we detrend the variables that we're interested in monitoring. And we use adaptive lasso in this case, and we've shown in additional studies that using adaptive lasso tends to work very well, and we don't have to spend a lot of time with the variable selection. It's just very simple, straightforward approach for detrending. We standardize the residuals, and then we select a time step in the variable where the shift occurs, and we are going to be using fused lasso in this case to look for shifts in the mean of the residuals. And then we identify the fault locations, the points in time where a fault's been identified, and those corresponding variables. So first step is to detrend the data. So this is looks fancier than it is. We're basically just constructing a model for the mean for each of our variables that we're interested in. And we call this the in-control model. And we're going to take this in-control model and plug in observations from the monitoring period in order to get residuals. So this is how we'll obtain residuals. And then we fit P linear regression models here on these residuals. And what we're interested in is, this is basically just an intercept model, right? We're interested in whether this intercept changes. So it's an over-parameterized model, clearly, because there are as many parameters as there are observations here. There's one mean for each observation. But that's why we use fused lasso, because it allows us to constrain the estimation and get estimates for these betas. So we observe a change in the mean of the process when beta I at time t is not the same as beta I at time t plus 1. Yeah, so this is a particular variable then is then classified as shifted at that time if there's a change point at that time. And we use fused lasso from the Gen Lasso package to do this. Well, so this is what fused lasso looks like. We've got our Z. These are our residuals. This is the mean from the model, and this is the constraint, which is constraining the absolute value of the difference between two consecutive means. And the key thing here is choosing this regularization parameter. And we're borrowing some of this methodology from Capizzi and Mazzarato's 2017 paper, and they used this extended BIC criteria to choose lambda, but they also assumed that their observations were independent. So what we have done is this tau represents the sample size, and so what we have done is we're going to adjust that sample size using a formula for the effective sample size. based on a fitted time series model to the residuals. And then we replace this sample size with this effective sample size throughout this EBIC criteria. So we'll choose lambda such that this adjusted EBIC is minimized. And for the sake of time, I'm going to skip forward and just say we do compare this method to Capizzi and Mazzarato's method, which is called M phase 1. And they are assuming IID data. And there are some similarities between our process and theirs, but they apply theirs directly to the data. We're going to apply their method to our standardized residuals that we have obtained. And we did a simulation study, but I'm going to spare you the details. It does work well across a variety of metrics, especially compared to the Capizzi and Mazzarato method. And we look again at this same case study. So we've got this 10-minute data with that known pH fault. And we have in control data, about 1,200 observations and about 1,000 observations that we're going to monitor. And we wind up with 20 variables that we are going to monitor. So dissolved oxygen, pH, conductivity, total suspended solids, etc. And we've sorted out explanatory variables that we'll use to detrend. So these are things like temperature, the blower flow, the flow of oxygen by the blower into the SBR, various state variables that says whether particular blowers are on or off. We put in lagged values of the control variables because sometimes the impact of the variable now isn't realized until a little bit later on. And we do know that there are diurnal components especially in characteristics of the influent wastewater. So we plug in some harmonic components to account for these daily, 2-hour, or hourly trends. And so we fit these models with adaptive lasso. So across the top, these are our 20 monitoring variables. And down the side, these are the explanatory variables that we use. And you can see these are all coefficients from an adaptive lasso model. So it automatically will sort of remove any of these diurnal hourly and 2-hourly components if they're not needed. And then we can identify those variables that are important. And so this is what those fitted trends look like. So just as an example, this is return activated sludge temperature. So you can see the black is the true data and the red is the predicted value. And so then these are the residuals, and there's this big drop. This area to the right of the blue dashed line is the testing period. So you can see there's a big drop in the RAS temp here. Similarly, in the level of the tank in the MBR, there's a big drop down in the testing period here that is not accounted for by the model. So you can still see it in the residuals. These are the kinds of changes that we want to be able to detect. And we get our effective sample size. Now we have 20 monitoring variables, so each one of those potentially could have a different effective sample size. So we looked at all of them and we fit an AR1 process to all 20 of them because that captured the autocorrelation pretty well across all of them. And then we took the lag one autocorrelation from each of them and just averaged them across the P variables. And so you can see here that these are the PACF plots of a few of the variables. And so you can see that an AR1 model does fit the autocorrelation pattern pretty well. And so when we averaged across these 20 response variables, we had an average lag one autocorrelation of about 0.6. So we use that to adjust the sample size. And I'll show you the results on the next slide, comparing the Capizzi and Mazzarato in phase 1 against our method's results. But before I do that, I want just to kind of orient you to this particular figure. These are the residuals of dissolved oxygen across this time period. And this shaded green region is where we suspect that pH fault occurred. And so when you see an arrow, that's an indicator that a shift in the mean was detected in this particular variable, and in this case, it was a shift downwards. So the downward shift is detected here, and an upward shift is detected here. And the size of these arrows, we categorized into small, medium, and large. So if it's a large arrow, it's an indicator that there was a large shift that was detected there. So in this first column, what we have are the results from Capizzi and Mazzarato's in phase 1. And in the second column, this is our method. So the first thing to notice, and I haven't shown all the monitoring variables here, just a subset of them, is that in phase 1 identifies 94 faults, and we identify 56. That was one of the characteristics that we noticed in phase 1, is that it identified a lot more faults than our method did. And we want to avoid false alarms as much as possible, because operators really don't like the boy who cried wolf. They will begin to ignore these alarms that are issued by monitoring systems if they come too frequently and erroneously. A second thing that we notice is when conductivity is going up here, and in phase 1, we get some conflicting information. Sometimes it's detecting that conductivity is going down, then it goes back up, then down, then up again. And the same with pH. While we know that pH is just continually going down, what we're seeing here is that sometimes in phase 1 is detecting that it goes up as well. But over here, with our methodology, we have very consistent upward trends for conductivity and downward trends for pH. So this is also a positive indicator for us that this methodology is doing what we expect it should. So what was the problem? What happened is in Colorado, they where this system is located, of course, it snows in the winter, and they put salts on the road for traction. And there was a big spring rain in this time period between April 21st and 24th. And so all of that salt gets washed off into the waterways. It goes into rivers and streams and into reservoirs. And when that water is ingested by a conventional water treatment facility, they don't remove salt. They're not designed to remove salt. And so those salts actually pass through to the distribution system. So the water that people were turning on in their homes was saltier than usual. And so that's why we see this conductivity was increasing during that time period. At the same time, the precipitation diluted the alkalinity in the surface water, which reduces the water's ability to buffer against these changes in pH. So as the pH began to drop, the microorganisms began to die off, and then it became a bit of this circular issue where it was in a downward spiral and couldn't be saved without intervening at that point. So that's what happened. And I'll say that there's so many other projects. I don't have a lot of time left, but there's a lot of other projects that over the past 10 years, Zahi and I have worked on together. And every single different system has different inputs, different outputs. And so it requires understanding of all these systems. So I've gotten a crash course in some of these engineered systems. And every single one has different interesting things that can go wrong with them. So with just my last couple of minutes, I'll just very briefly scroll through a couple of other interesting projects that we're working on right now that have graduate students working on. So one of my students is working on looking at detecting changes in variance, because in systems that are controlled, the idea is that the mean doesn't change. So in this particular system, this is a forward osmosis system, the flux across the membrane is controlled to stay the same, but if there's any sort of fault that occurs, the variability of that particular controlled variable will begin to change. And so we want to be able to detect when the variance changes or when the covariance between two variables changes. And And then, of course, when you start talking about monitoring covariance, there's lots of ways to do that. But the covariance matrix can get very, very large. So even say with 5 variables here, this becomes a 5 by 5 matrix. And sometimes not all variables are highly related to others. And so some of them might just be 0, and we want to kind of sparsely estimate this covariance matrix. So we're working with Rakon Kim, who's also a graduate of A&M and is now an assistant professor here at Baylor, with one of the methods that he developed for positive definite thresholding of covariance estimators and incorporating those into a methodology for monitoring covariance. And so far have positive results in being able to identify quickly shifts in variances and covariances. We've also worked with a lot of data from this direct potable reuse trailer. So you can see the trailer here. The idea is that this trailer can be attached to the back of a truck and transported anywhere, and can receive effluent wastewater, treated effluent wastewater from a wastewater treatment facility, and treat that water such that it is ready to be consumed so that it's potable. And there are 6 different systems inside of this trailer that you can see here. And one of the important systems is a microfiltration or ultrafiltration component. It's way in the back of the trailer. And what we see is that over the course of about 2 weeks here, anywhere from, I think this is 10 days to about 2.5 weeks, we'll see that the transmembrane pressure, the pressure on the membrane in the ultrafiltration unit, it increases. So there's kind of this global trend. And then every hour, the membrane is backwashed. So there are also these local trends that you can see here. And then there's variance across these cycles as we move across this entire run, and there's variability within the cycles. So we're working on building models to capture all of these differences. And so here's real data from the system, and then this is simulated data from the system. So we've been able to capture a lot of the same characteristics of the real data. And these are functions. So basically each one of these guys gets split up between backwashes, and we can plot them and overlay them. And so we can see how these functions, the magnitude of these functions is changing over time. And we can also use these functions for identifying faults. So we can monitor the functions themselves. So we used a paper and a methodology presented by Dye and Jenton in 2019 to capture both a magnitude score and a shape score for each function, and then we can monitor those over time. So I'm almost out of time, so I'll skip past this one, but this is pretty exciting stuff because functional data analysis has never been used in this field before, and so people are really excited about adapting some of these methods for the particular monitoring scheme at hand, so that we can monitor the whole function, because there are lots of water and wastewater treatment processes that wind up producing functions for their observations as opposed to just scalar values. So finally, just to wrap up, this is a public repository that we've developed on the Harvard Dataverse, where we're posting all the data sets that we're collecting from all of these projects. And if the project is also associated with a journal publication, then we have all of the R code and scripts needed to recreate the results in the paper posted here as well. And so there's a QR code there if you're interested in looking at any of those data sets or using those as a case study in your work. So those are publicly available now, and anybody can use those. And just to wrap up, those are some of the papers that we've published as a result of all of this work. And I'm happy to answer any questions now.
Does anybody have any questions for Dr. Herring?
Marcy, can I ask a question?
Absolutely.
Okay. Mandy, it is excellent talk. I learned some things, and it is sort of an example how the influential and impactful statistical research could be.
Yes.
I was affiliated with CDC dental group maybe about 10 years ago. The big thing that they were involved was the fluoridation, and they wanted to address the issue that in some areas where fluoride is naturally appearing, to have this adaptive fluoridation level.
Oh,
So
nice.
I don't know if you had any fluoridation or any analysis that involves fluoridation. It is a little bit controversial topic.
Yeah, I haven't worked with any fluoride data, but this idea of adaptively treating water is certainly a new one that's being proposed. So, for example, a lot of what people do is they'll add chlorine to water for its antibiotic.
Yeah.
Because when the water is distributed, you want it to maintain this, no bacteria in the water. So you add chlorine to kill all the bacteria. But chlorine, when it interacts with some constituents in the water, will produce carcinogenic compounds. So people are looking at peracetic acid as an alternative. But peracetic acid's more expensive. And so you don't want to dose more of it than you need to. So they're looking at one of the projects one of my former students worked on was looking at tracking E. coli. Can we predict E. coli levels in the water so that then you can specify the amount of peracetic acid to add to the water? And then, of course, there's lots of work on contaminants of emerging concern, pharmaceuticals in the water, personal care product derivatives that are in the water as well.
Thanks.
Yeah.
I think we've got a couple comments along the same lines, Dr. Vidakovich, and I also thought the same thing. This is a very, very fascinating subject, right? It's truly a real world application of impact of statistics in our environment, things that we need and use every day for our resources.
Yeah, and we don't think about water, I think, sometimes as much as we should, and it's been a really great area and rich area to work in, not just because of the impact that it has, but also there's so many great statistical problems that are present here that haven't been studied. So that I think is probably that intersection of a great problem with real world impact crossed with, no one's made methods for this yet.
Right.
Tends to be a really great place for an academic statistician to work.
Right. Some real true challenges, right?
Yeah, exactly.
Yeah. Very interesting. Does anyone else in the meeting room have any other questions for Dr. Herring or comments? Make sure we give everybody opportunity to speak up if they would like to. Looks like we have everybody. No further questions. It was very, very exciting topic. Very interesting.
Yeah, thank you. Thank you all for having me. It's fun to put this together.
Yeah, it's always fun to bring it all back to home, right?
Yeah.
Yeah.
And Marcy and Amanda, are the slides available? Can you share those?
Yeah, I'm happy
Yeah.
to share them. Shall I email them to Marcy or Elaine?
No, go ahead and send them to Anna or
Anna?
Elaine, and
Okay.
they will post them on a web page where we have all of the previous talks. Since you've already given permission for that, that should be no problem.
Yeah,
Especially Harvard database, it is interesting.
Yeah, I'm happy
Thanks
to do
for
that.
that.
Right. Well, thank you very much, Mandy. It was great to see you and talk to you in person, and to learn more about what you're doing and how you're sharing your knowledge and experience that you gained both at A&M and the other institutions in the real world. It really drives it all home. It's very nice. Thank you.
Yeah, thank you.
And I invite you all to participate in next month's webinar. Will be middle of April, I think April 10th. So Anna will be sending out an announcement in regard to that shortly, I guess, before too long. So have a great day.